vocabularies and synonym mappings and for all of this domain-specific reasoning. So that's where MICBO comes in. It's a lightweight, extensible ontology for organizing mammalian cell cultivation knowledge, and MICBO fills that missing layer between existing OBO ontologies and real-world process workflows, biological processes, these biomanufacturing processes. It enables machine actionable understanding so that computers can finally understand, they can know what I mean when I say fed batch culture with glucose limitations. So MICBO is an application ontology, meaning that it's designed for a specific practical domain. Here, the domain is mammalian cell cultivation, but it's anchored to more general upper-level ontologies. So MICBO is authored in OWL2 and developed in Protege. We all know what that is. And at the heart of MICBO is the central class cell culture process, which branches into subclasses batch, fed batch, perfusion, and chemostat. And the philosophy was to build a minimum viable layer precise enough to capture the essential concepts and yet flexible enough to extend and grow with the data. So this is a deliberate restraint that keeps the ontology deployable in industrial settings where overmodeling it could deter adoption. So we're adding a SCOS layer to manage the synonyms and make the ontology vocabulary user-friendly for scientists and interfaces. And again, the goal is a practical first step towards fair data and machine-readable experimental annotation in this domain. So MICBO follows the hub and spoke model, extending the industrial ontology foundry, or IOF ontology, into the biological manufacturing space. Its modules describe cell lines and process parameters, media and outputs, such as productivity and titer. Measurements like cell viability, productivity, gene expression, all specialized OBO term measurement data, bringing in standard units from the units ontology and descriptors from OBI and SBO for consistency. Shared object properties enable both top-down reasoning and bottom-up data integration, supporting scalability while allowing academic and industrial data sets to coexist without redesigning the schemas for each newly introduced data set. MICBO is implemented using a lightweight Python-based turtle workflow. So this setup allows us to evolve the ontology quickly as new concepts and relationships emerge, while also supporting an automatic generation of metadata web forms or templates for data entry that are consistent with the schemas and the ontologies. For term discovery, we used a hybrid approach combining manual review of subject matter experts with LLM-assisted literature mining. And the language model helped to service common metadata patterns across papers and reports and databases, all this unstructured data. For example, the recurring trio of pH, dissolved oxygen, and temperature, which was consistently defined across different culture conditions. So by capturing these recurring parameters as shared ontology classes and relationships, MICBO provides a foundation for consistent machine actionable metadata across bioprocess datasets. We validated the MICBO ontology in Protege using both the structural view and Hermit Reasoner to ensure internal consistency. And the ontology successfully materializes expected inferences, particularly across the cell culture process hierarchy and associated measurement classes. All class and object property axioms were found to be logically consistent with no unsatisfiable subclasses, and the reasoner confirmed coverage across all the competency question targets, including cell culture parameter groupings and process subclass distinctions. So in short, the model is coherent, inferentially complete for its current scope, and stable for integration with OBO-aligned external ontologies. To test the coverage, we manually curated 724 RNA-seq bioprocess runs from published CHO studies. And of these, 518 were batch, 135 fed batch, 49 perfusion, and 22 were not classified by any subclass. We couldn't figure out which subclass it was using from the unstructured data. We modeled and tested 5 competency questions, including the 3 here that are representative of our main classes of questions, which culture conditions correlate with high productivity, which cell lines overexpress a particular gene, what pathways differ from between the different subclasses. And then we asked whether or not these different CQs could be expressed and answered within the ontology. MICBO did successfully capture the relevant relationships for the three main classes of CQs, which were the culture optimization, cell line engineering, and process comparison. All queries executed in sub-second time, showing that semantic reasoning scales efficiently, a practical alternative to inferencing from LLMs, which have high latency and also brittle multi-table SQL joins. So the ontology harmonized the heterogeneous metadata into a single knowledge graph, enabling reproducible cross-study comparisons. MICBO aligns the OBI, UO, SBO, and IAO ontologies. We're using units, process types, and metadata conventions. This makes the data fair, findable, accessible, interoperable, reusable, and the data harmonized under MICBO can be automatically checked for completeness, linked to external ontologies, and exposed to AI pipelines and web APIs, which we did because MICBO provides the semantic backbone for our application iBioHub, where scientists can explore bioprocess data through natural structured questions. For example, show the cell line fed batch runs with glucose limitation and high antibody yield, for example. Behind the scenes, AI pipelines extract and structure the metadata from legacy notebooks and papers and all these unstructured data so that they can be rendered in web forms and allow the authors to validate the data instead of re-entering it in a structured format. This approach makes experimental data discoverable by concept, not just by keyword, supporting reproducible analysis, cross-lab comparison, and next generation of digital bioprocessing. MICBO provides the semantic specification that enables the automated ingestion, transforming legacy unstructured bioprocess data into machine-readable knowledge, and that foundation powers our iBioHub, a web-based platform that connects data, knowledge, and tools across mammalian cell biomanufacturing. So iBioHub serves as a centralized hub for integrating omics, genomics, and process data through a shared semantic layer, ensuring the standardization and interoperability across labs. On top of that, AI-enabled analytics automate annotation and optimization, while the platform's secure, scalable architecture supports the community-driven collaboration and fair, reproducible biomanufacturing data. Okay, so here is a demo to show you a little bit about what this is all enabling. This shows the iBioHub conversational-like interface, which will operationalize this MICBO ontology. So, for example, a user will be able to issue a natural language request, for example, show fed batch, show S transcriptomics colored by productivity. And then the system uses an LLM grounded in MICBO to interpret those entities and relationships with AI agents enabled with tools for translating the requests into Sparkle and property graph queries executed across both RDF and NoSQL stores. So the results can be drawn from iBioHub Knowledge Graph, where instances and metadata are annotated using MICBO classes and object properties, and a reasoning layer can validate type constraints and ensure semantic alignment before visualization. So this demonstrates AI-assisted ontology mediation in practice, combining that natural language understanding with formal reasoning to make bioprocess data findable, interoperable, and interpretable. So our next milestones include formal mappings from MICBO to IOF core and BPOG, BPSA, ISPE standards. We intend to register at identifiers.org and BioPortal and pursue OBO Foundry acceptance. And we're adding the SCA synonyms that I said before for vocabulary alignment. We'll also expand into multi-omics integration, including proteomics, metabolomics, and introduce semantic links to CHO genome scale models. This will allow queries that connect cultivation conditions with metabolic flux predictions, and that's going to enable digital twin and AI simulation workflows. So that's really exciting. So all this leads naturally to other AI-enabled work, including what we already talked about, the AI-enhanced pipelines for metadata extraction and validation. So as we wrap up, I want to highlight the broader impact of the work. MICBO and iBioHub together form the connective tissue between data, semantics, and discovery, translating the bioprocess knowledge into actionable AI-ready foundation for biomanufacturing. So this isn't just an ontology project, it's about automating intelligent, reproducible experiments at scale. All the files are open source. In summary, MICBO provides a principled yet pragmatic semantic framework for mammalian cell bioprocessing, bridging the gap between experimental reality and machine actionable knowledge and paving the way for fair, AI native, and interoperable data structures. And I'm out of time, but I just want to acknowledge my co-authors and Anne Rochelle from Sartorius, who spearheaded the public-private partnerships informing the strategy and direction of the science drivers behind this work. Thank you, and if there's time, I'll take a question. Vocabulary